Welcome to this overview of proteomics, where we explore how proteins reveal the inner workings of biology. In this presentation, we'll walk through core concepts, common techniques, quantitative methods, applications in medicine and industry, current challenges, and future directions to give you a clear sense of why studying proteins is essential for modern biological research. This slide shows the contents we will cover, mapping out the journey from introductory concepts to practical case studies and conclusions. Think of it as our roadmap. We'll start with the basics, dive into techniques and quantitative approaches, look at real-world applications and challenges, and finish by highlighting emerging trends and takeaways for future research. Let's begin with an introduction to proteomics and why it matters. Proteomics is the large-scale study of proteins in cells and tissues, focusing on their structures, functions, and interactions, and how these change in health and disease. Understanding proteins gives us a dynamic view of biology that complements genetic information and helps explain cellular behavior. Here we define the scope and context of proteomics, tracing its rise from protein chemistry to a mass spectrometry-driven field. Proteomics emerged in the late 20th century and now supports deep biological insight, enabling researchers to link protein changes to cellular processes, disease mechanisms, and potential therapeutic targets, illustrating its growing importance in biomedical research. Key concepts in proteomics include what proteins do and how their expression is regulated, plus how the proteome differs from the genome. Proteins perform diverse roles from catalysis to signaling, and their levels and modifications reflect cellular states. Unlike the static genome, the proteome is dynamic, revealing temporal and environmental changes not captured by DNA alone. Now we'll move into the main techniques used in proteomics and how they help researchers separate, identify, and quantify proteins. This section highlights practical laboratory and analytical approaches, showing how different methods work together to translate complex protein samples into meaningful biological information that can guide experiments and clinical decisions. Protein separation is a crucial first step and can be achieved in several ways. Gel electrophoresis, including SDS-PAGE and two-dimensional gels, separates proteins by size and charge. Liquid chromatography fractionates proteins based on interactions with a stationary phase. Together, these methods reduce sample complexity and prepare proteins for downstream identification by mass spectrometry. Identifying proteins relies heavily on mass spectrometry and complementary platforms. Mass spectrometry provides peptide mass fingerprints and fragmentation patterns for precise identification, tandem MS improves sequencing accuracy, and protein microarrays enable high-throughput detection and quantification, allowing researchers to profile thousands of proteins in parallel for discovery and validation studies. Quantitative proteomics focuses on measuring how protein abundances change between conditions and over time, allowing us to move beyond presence or absence toward precise comparisons. This section outlines strategies to obtain accurate protein quantification, the importance of experimental design, and how quantitative data support biomarker discovery, functional studies, and therapeutic development. There are several common approaches to protein quantification, each with pros and cons. Label-free methods compare signal intensities across runs and are flexible and cost-effective. Isotope labeling introduces stable tags for more accurate relative or absolute measurements. Metabolic labels like cyclic and isobaric tags like ITRAQ enable multiplexing and robust comparison across samples. Data analysis is where proteomics results become biological insight, relying on bioinformatics tools, statistical rigor, and network interpretation. Proper normalization and replication reduce variability, computational tools identify differentially expressed proteins, and integrating protein-protein interaction networks helps reveal key pathways and hubs that drive cellular responses and disease phenotypes. Next, we explore the many applications of proteomics across clinical, industrial, and environmental fields to show how protein-level data translate into real-world impact. From identifying disease biomarkers to improving crop traits and monitoring ecosystems, proteomics offers tools to solve practical problems and to develop new diagnostics, therapeutics, and sustainable technologies. In clinical and medical settings, proteomics accelerates biomarker discovery and therapeutic development by revealing proteins linked to disease states. This enables early diagnosis, target identification for drugs, and personalized medicine approaches that tailor treatments based on an individual's protein profile, improving prognostic accuracy and informing more effective clinical strategies. Proteomics also has strong industrial and environmental uses, from enhancing agricultural traits and food safety to monitoring pollutants and optimizing bioprocesses. By analyzing protein changes in organisms and ecosystems, proteomics helps improve crop resilience, detect environmental stressors early, and guide biotechnology efforts to develop efficient and sustainable production systems. Having seen the power of proteomics, it's important to acknowledge current challenges and look ahead to future directions that will shape the field. Limitations in sensitivity, data complexity, reproducibility, and sample preparation still restrict what we can measure, but technological and computational advances are steadily addressing these gaps and opening new opportunities. Technical limitations in proteomics include difficulty detecting low-abundance proteins, variability introduced during sample preparation, and the complexity of large proteomic datasets. These issues complicate reproducibility and interpretation, underscoring the need for better experimental standards, improved sensitivity in instrumentation, and advanced computational pipelines to extract reliable biological meaning. Emerging trends in proteomics point toward exciting possibilities. Single-cell proteomics promises insights into cellular heterogeneity. Integrating proteomics with genomics and metabolomics will enable richer systems-level understanding, and ongoing mass spectrometry advances continue to improve sensitivity and throughput, all of which push the field toward more precise and comprehensive analyses. Let's look at case studies that demonstrate how proteomics actually changes research outcomes and patient care. By examining focused examples in cancer and infectious diseases, we can see how protein profiling uncovers biomarkers, therapeutic targets, and mechanisms of disease, translating molecular measurements into actionable knowledge for diagnosis and treatment strategies. In cancer research, proteomics aids identification of biomarkers and tumor profiling by comparing protein expression between healthy and cancerous tissues. This differential profiling reveals candidate diagnostic markers and therapeutic targets, helps stratify patients for personalized treatment, and deepens our understanding of tumor biology and mechanisms driving disease progression. Proteomics in infectious disease research examines pathogen protein landscapes and host pathogen interactions to reveal virulence factors and immune responses. Analyzing pathogen proteomes identifies drug targets and vaccine candidates, while profiling host responses uncovers defense mechanisms and biomarkers indicative of infection severity or treatment outcomes, informing better therapeutic strategies. As we wrap up, this summary highlights the main points. Proteomics provides a dynamic view of biology. Powerful techniques enable protein separation, identification, and quantification, and applications span medicine, industry, and environmental science. Although challenges remain, continued innovations promise deeper insights and more direct translation of protein-level data into practical solutions. Key takeaways are that proteomics is essential for modern biological research, enabling biomarker discovery, therapeutic development, and systems-level understanding, while technological advances in mass spectrometry and bioinformatics are driving its impact. Keeping rigor in experimental design and data analysis ensures reproducible, meaningful results that can advance science and improve human and environmental health. Looking forward, proteomics will expand the proteome landscape and integrate more closely with other omics to build comprehensive models of biological systems. These integrative systems biology approaches will enable personalized diagnostics, new therapeutics, and a deeper understanding of complex interactions across molecular layers, setting the stage for transformative discoveries in the years ahead. Thank you for watching this journey through proteomics. Your interest supports progress in a field that bridges molecules to medicine. If you want to explore any section in more detail, from specific techniques to case studies, I'm ready to help you dive deeper and translate these ideas into experiments, analyses, or practical applications.